All right, we're down now to what I would consider our last week of class. So, you typically, in the last week of class, we take a look at some clinically interesting viruses and viral diseases. So, with this lecture, we're going to begin our double stranded DNA, and we're going to take a look at the family Herpesviridae right there. Let's see, we're also going to be covering this Orthomyxoviridae, that's the influenza virus. HIV is this Retroviridae over here, and down here is our Coronaviridae. So those, just a sampling of a few different viruses we're going to look at more in depth. I just wanted to review a little bit about viruses in general, how we classify them, for instance. So remember, there's two different ways that we classify viruses. That hierarchical virus classification is when we try to kind of squish virus particles, right? They're not technically living, at least most people don't consider them to be. We try to fit them into our classification with living organisms. So with this system, there are, I think, a few classes and a few orders, but all the viruses then are fit into families, which that's the level of taxa that will be kind of considered with family on down. Remember, the viral families end in viridae, and as you'll notice over here, they are italicized. Everything from the family on down is italicized if you write them correctly. And then viral genuses are always going to end in the suffix virus, like herpes virus we'll be talking about today, and that's also italicized. Okay, and then just remember that other classification system, the Baltimore classification, we've got, and that looks like this graphic doesn't talk about that at all, but this is all based on the nucleic acid, the kind of nucleic acid they have, whether it's single-stranded DNA, double-stranded DNA, single-stranded RNA. There's 7 different types, so group 1 through group 7, and I wouldn't worry about which is which. Those are just the 2 classification systems that we see. Okay, just a little other kind of basic review, viral terminology here. Remember, if a virus does not have an envelope, we call it a naked virus. And here you can see the capsid. That's made out of protein capsomeres. Here, for example, this is an icosahedral capsid. It's a polygon with 20 different sides. That's probably the most common shape of capsid because it's very stable, very symmetric. It's going to be protecting some form of nucleic acid, whether that's DNA or RNA, never both, one or the other. And then here's an example of an envelope virus. We've got that nice envelope surrounding the capsid. Remember, most animal viruses do have an envelope, viruses that infect animals, humans. They often, not always, have some kind of spike, maybe glycoprotein spike sticking out from that envelope, or you could have those sticking out from the capsid as well. Usually going to be helpful for recognition, adherence, that type of thing. Okay, so just some quick review terminology. Let's zero in on some characteristics of the Herpesviridae, family Herpesviridae. All right, this is a double-stranded DNA virus. So chapter 24 is just examples of double-stranded DNA viruses. We are only going to cover the herpes virus. There's some other ones in there, papillomaviruses, whatever, if you want to read about those. You're welcome to. This is a pretty large family of viruses, so we're going to talk about actually 8 different genera of herpes virus, but a little general information first. I'll point out in this graphic here, they call it Herpes Simplex Virus. That's the old name for it. now we call it HV for human, herpes human virus instead of herpes simplex virus. You still see this HSV quite a bit. I'm not sure if the name change is just really new or whether it's just kind of hard to change once you're used to calling it something. The new name hasn't caught on yet, and I'm not really sure. All right, so characteristics of this virus. It is enveloped. Like I said, most viruses that infect humans are, or a lot of them, not all of them. The capsid is icosahedral. Okay, as you can see from the picture there. And it's a double-stranded DNA virus with a linear genome. Okay, so that means it's not chopped up into segments. It's all, like you can see here, over here, it's all one strand. And it's a pretty large genome, somewhere between 80 and 100 genes. So if you compare that to the virus that has the smallest amount of genes, hepatitis B only has 4 genes. So pretty large in terms of its genome. It does have a prominent feature that we call the tegument. So you can see that's over here. It's kind of the purple circles there in this graphic. The Herpesviridae are not the only viruses that have a tegument, but it's quite prominent in Herpesviridae. What is this? These are proteins that it carries with it that are going to suppress host immunity. And an example, some of those proteins actually suppress interferon alpha and beta production. So that's type 1 interferon. Remember, that's our innate system's kind of response to virally infected cells. All right, so these tegument proteins suppress our host cell production of that type 1 interferon. The other thing that they do, just in general, aid in viral replication. Okay, and there's about 20 different proteins that it actually carries with it. All right, you'll notice that it also has some surface spikes, glycoprotein to be specific, so sugar protein spikes. And their job is to recognize, bind to, and cause fusion with the cytoplasmic membranes of the host cell. Okay. What else can we say in general about the Herpesviridae? Well, they're pretty widespread. Estimates that I've read, as much as 100% of us have been exposed to human herpes virus in some form. Okay, they have and it doesn't have to be, a person doesn't have to be in contact with an active lesion in order to get the virus transmitted. It's more likely to be transmitted by an active lesion, but sometimes it can be passed anytime. So very high infection rates. It's possible that you've had a subclinical infection. In other words, you didn't have any symptoms, but you probably have antibodies to herpes, probably have been exposed. What's the reservoir? In nature, well, it's humans passing it around to other humans. There are herpes virus that's specific for animals, but we don't think they cross species, right? So herpes is not considered zoonotic, okay? Another thing about all the herpes virus is that all the genera, we'll say all the members of the Herpesviridae show latency. Okay, so they don't do just the lytic cycle. They do what we call lysogenic cycle in bacteriophage. Okay, in animal viruses, we call that latency. And they go latent, they actually go latent in the form of episomes. Okay, so you'll see this. This is a great 2-3 minute video animation that shows kind of what happens, how the virus gets into a cell and how it leaves it again. It's a great animation, but you'll see that the double-stranded DNA, let's see, if here's a, let's say this is a bacterial cell, and here's an animal cell. Okay, remember when the bacteriophage integrate into that host chromosome, remember round chromosome for bacteria, okay, that's how bacteriophage go latent. Okay, but animal viruses, let's see, here's the nucleus. Remember, our chromosomes they're linear. Typically, the Herpesviridae forms kind of a little plasmid-like structure that we call an episome. Okay, and that's considered integrating in because that little piece of DNA is going to get replicated whenever the cell gets replicated. Once it's latent, it won't ever come out again. Okay, so this again is a plasmid-like circular DNA. Okay, and again, that's inside the nucleus. But again, you'll see that if you watch this little animation. That's a nice animation. Some of the Herpesviridae are oncogenic. So they have the potential to form cancer. There's some environmental factors that play a role there, as well as a person's genetic predispositions. And in terms of its life cycle, I'll put life in parentheses because it's the particle. We don't really consider it alive, but the viruses replicate and assemble in the host cell nucleus. And then they pull their envelope from the nuclear membrane. And that's kind of unusual. Okay, again, you'll see that in this animation. Hopefully you watch that. And then how do they leave the host cell? They exocytose. We'll say exocytosis, or they lyse from the cell. Okay, more often it's going to be a little bit slower exocytosis, so they're actually kind of budding off when they leave the cell. But sometimes when they do this so quickly, it accomplishes the same thing. It lyses the cell if you have so many virus particles doing that all at once. Okay, what are some of the effects of herpes virus disease? We're going to look at some of them in a little bit more depth, but we can say a little bit about what all the different genera have in common. Okay, in general, they're all going to target the mucosal membranes. Okay, so a little cut or break can allow the virus particles in. Think about our lips. How often in the winter are your lips cracked a little bit? Okay, really nice portal of entry there for the virus. Okay, the virus are going to multiply in the basal and epithelial cells in whatever region we're talking about. Okay, basal cells are just the deepest layer of the epithelium, and epithelial cells just line all the cavities and surfaces of structures throughout the body. Okay, so this is going to cause localized inflammation. Okay, and probably some cell death. Oops. Okay. All right, and the other really kind of interesting thing we see a particular cytopathic effect. Okay, we abbreviate that CPE, cytopathic effect. So we often look for cytopathic effects of viruses as a way to diagnose the infection. Cytopathic effect refers to virus-induced changes in our cells visible by light microscopy. Okay, we very rarely diagnose a virus infection by electron microscopy. It's just too time-consuming, too expensive in order to do that. So instead, we take a sample of host cells and look to see how those host cells might have been changed by any virus infection. And a particular cytopathic effect that is not unique to herpes, but it's very prominent in herpes infection, is the formation of syncytia. Okay, and this just means multinucleated cells. Okay, so in this picture here, we have a nerve cell, and all these little dark spots here are nuclei. So we have a nerve cell that has multiple nuclei. Not sure why some of them are outside of the cell here, but think about it. This is a very ingenious mechanism that the virus has to evade host detection. All right, when the virus particles are outside of the cell, they're quite vulnerable to neutralization by our antibodies. Okay, so the strategy is for one infected host cell to just fuse with another cell that hasn't been infected. Okay, and in that way, it's spreading the virus particles from cell to cell without ever being outside of the cell. Okay, herpes virus is also able to downregulate MHC type 1 expression. All right, so we don't get good help from cytotoxic T cells because this is not advertising by those MHC class 1 that it's infected with something. Okay, so here's just another picture of, again, that cytopathic effect. Here's a multinucleated cell here. And again, we call that syncytia. So I want to give you an idea of how big this human herpes virus family is. Okay, we are going to focus and spend a little bit more time on the first 4 genera here. So HHV 1, 2, 3, and 4. All of the 8 human herpes virus go latent, but they go latent in different kinds of cells. So, for instance, HHV 1, 2, and 3 all go latent in nerve cells. HHV 4 goes latent in B cells, as well as HHV 8. And these 3, 5, 6, and 7 go latent in stem cells. Okay. All right, so we're going to spend a little bit more time on HHV 1 and 2. HHV 1 is also referred to as simplex virus. This is going to most commonly called cold sores. So sometimes we refer to that as above the waist herpes. Simplex virus, or HHV 2, is going to cause the genital sores, below the waist herpes. Varicella virus, or sometimes we refer to that as varicella zoster virus. This is the causative agent of chickenpox, and that can reactivate as shingles. That's always an interesting discussion. And HHV4 lymphocryptovirus, that's also called Epstein-Barr virus. And that can cause a couple different infections. The one that you're probably most familiar with is mononucleosis. All right, so we're going to talk about all four of those in a little bit more depth here with the next video. I wanted to just briefly mention the other members here. Cytomegalovirus is also, we abbreviate that CMV. That's quite widespread, but almost all the time it's asymptomatic. So people don't even know they've been infected with it. Sometimes young babies will have symptoms, or AIDS patients that have very poor immune response might show some symptoms. HHV 6 and 7th, the 6 causes roseola, which is a high fever. It's typically presents as a high fever accompanied by a bright red rash. Usually very young children get that. Typically don't have to treat it, it just passes. And the HHV7 is what we call an orphan virus. We don't really know of that causing any disease, okay? So as far as we know, that doesn't cause anything. Excuse me. And then HHV8, radenovirus. This is another, it's one of the herpes virus that can be oncogenic. It basically, it causes this Kaposi sarcoma associated illness, which basically causes tumors only when the host is super immunocompromised. The virus actually targets a tumor suppression pathway. So we started to see lots of cases of this showing up in the early 80s, and that was a big tip-off that there was something else going on there. We see this in association with HIV and AIDS. Okay, so that's a little overview of the herpes virus. Again, next video we'll go on to HHV1 and 2 for sure, and probably the next video 3 and 4. Okay, thanks for listening.